Good afternoon, everybody on shore, or whatever time it is for you at home, wherever you are joining us from. This is Research Vessel Falkor, ROV SuBastian. My name is Mardi McNeil, and I'm a marine geoscientist from the Queensland University of Technology in Brisbane, Australia. And you're joining us for the first dive of the northern depths of the Great Barrier Reef expedition. It's an absolutely glorious day out here today. I hope it is at home where you are too. We're situated at the moment about 65 nautical miles northeast of Fraser Island off the Queensland coast, Australia. And today we're going to be doing a midwater plankton survey. Oh.
Siphonophore gilmeri.
And it's all happening already. We're going to be joined today by Dr. Dhugal Lindsay, who is joining us remotely via satellite from the Japan Agency for Marine Science and Technology Centre. Dhugal's not on board the ship, but we are collecting high-resolution video imagery of the water column plankton and soft-bodied jellies, which is Dhugal's specialty. So I might hand over to Dhugal to introduce himself, and Dhugal's going to take us through the dive and talk to us about what he's seeing and what we're observing and what's going on in the water column. So I'll hand over to you, Dhugal.
Thank you, Marty, and a big thank you to Schmidt Ocean Institute and everybody there on the Falkor helping to make this happen. As Marty just said, I'm an Australian scientist who's working here at the Japan Agency for Marine Earth Science and Technology, JAMSTEC, and my speciality is deep sea plankton. And I was unable to be there on this cruise due to coronavirus travel restrictions, but I'm very happy to be able to take part remotely like this, seeing in real time all of these wonderful animals. This is the first time there's been a midwater ROV dive. This is Fraser Island. We're diving off here. This is the first time south of the Capricorn Channel, I believe, that anyone's ever seen what's in the midwater off the Australian coast. So really looking forward to seeing what we can see.
Hey, I didn't know that. That's awesome.
Yes. Another first.
Another Falkor
just before
first.
going. Yeah, what's that? A ctenophore, maybe. We had a siphonophore, Phistophora gilmeri, come through the video just as we started streaming before. You may have heard me yelling out excitedly. It's always great to get a positive species ID on these animals so that we can see exactly what habitat they live in. So we now have a time, latitude, longitude, depth, salinity, temperature, environmental reading for where that animal occurred. Ooh, what's that? Ooh, I think that was probably Collidarian radiolarian colony, donut-shaped. We should be pretty much out of the photic zone now where photosynthesis is possible. So all the plant plankton won't be actively producing energy and body mass. And all of the food that we find down here will either be sinking from the surface or it will have swum here actively. Down at depths like where we are now, we're at 232 meters. Around about here, possibly a little deeper, we'll start to see whale food. There'll be krill, euphausids, which, hello, what do we have here? This looks like a siphonophore. The transparent bit is where the swimming bells are. The whitish bit is the siphonosome or tail. It's a Physonex siphonophore because we can see that bright white spot at the tip of where all those clear swimming bells are. And if we get a bit closer, we may be able to tell what it is. It's very hard to move the ROV around at these shallow depths because we're constrained by the tether pulling on us, and the current is pretty strong. So it's a very challenging dive probably today for the pilots. This looks, oh cool. So this is the other Physophora species. Physophora gilmeri has reddish palpons. This one, the palpons seem to be mostly transparent. So it's the second species of hula skirt siphonophores, Physophora hydrostatica. So now we can say, well, gilmeri was a little bit shallower, but this other species occurs here also round about the same depth and environment. So somehow they are managing to coexist even though they're closely related species. Possibly they eat different food, or they've got some other way to apportion the niche space. And here is a chitignath or arrowworm in the foreground, which would provide good food for something like a siphonophore. So we're also able to get information not just on the physicochemical environment, salinity, temperature, etc., but also on the possible food, prey animals. And if we're lucky as well, we might see some fish or other predators. Nice one. Oh, yes. Beautiful. Well done. That's a money shot. Yeah, very, very long palpons, those long sausage-like structures coming out from the base of the animal there. They're palpons, which are the sensory zooids. A siphonophore is made up of a group of zooids, or like, I guess they're like internal organs or organs on a human, like the heart or the liver or whatever, except they're a lot more autonomous than our organs. So you couldn't take out your heart and leave it in a petri dish for days. It'd die pretty quickly. Whereas a siphonophore zooid, because each of them is a separate polyp, you can remove them from the animal, and until they use up their energy reserves, they'll quite happily live there in the little bottle of water you have them in. If you remove one of the stomach zooids, for example, with its attached tentacle, you could feed it krill or something else quite happily in a petri dish, and it would go on eating and living for weeks until possibly fungus or bacteria or something overcame its defenses and it might die. Things like the swimming bells, they'll swim around until they run out of energy, and then because they can't swim, they'll normally sink down to the seafloor where they're eaten by animals waiting there for the fall of marine snow from the shallower layers. Not just the seafloor, of course, but any animals who live in the water column below them will quite happily munch down on things that are sinking out of the water column. We've got multiple cameras here on the ROV SuBastian. This is the downward-looking camera that I'm seeing now, and you should be seeing soon. We have a 4K pan-tiltable camera for zooming in on all of the animals, and other cameras that are fixed focus to allow the pilots to see where they are when somebody's zooming in on an animal. They're still able to see where it is, so they can get closer enough to sample it without blowing it out of the way using the thrusters of the ROV. And so there's a range of different cameras laid out here on the SuBastian so that we can look at every part of the water column and the sea floor and record them all simultaneously. (242 meters depth). Another siphonophore. These siphonophores, because they're a colony of polyps, a colony of zooids that are all living together, attached to a single zooid that acts like the pipe through which nutrients are sent around to all the zooids, and that gather the wastes for excretion through this single zooid that acts as a pipe, kind of like an artery or a vein, I guess. And all the other zooids attached to that. So rather than like normal jellyfish polyps, which might attach to rocks or wood or shells, these siphonophore zooids attach to each other. And they swim around or float around as a colony, a polyp colony in the midwaters. Some of them can get very large very long. Actually, the longest animal in the world is a siphonophore. It's not a male blue whale, which reaches about 20 meters in length. It's rather a siphonophore, which can reach over 40 meters in length, twice the size of an adult blue whale. Looks like a string of marine snow. Siphonophores, being long and thin, look a lot like some of the particles that are produced in the upper layers where there's lots of food, and as they sink down through the water, their cross-sectional surface area becomes less, making it easier for them to sink, and they tend to become long and string-like as they fall deeper and deeper in the water column. So it can sometimes be pretty hard to tell the difference between a sinking particle that's extended itself out like a string and a siphonophore. And there's a lot of both of those. There's also a lot of little transparent animals I'm seeing on the video, which are probably either filter-feeding doliolids, which are a kind of a sea squirt, or little siphonophores as well. Quite a lot of marine snow here. I was surprised on the last cruise that we did up around the Great Barrier Reef. The amount of marine snow on some of the dives was really phenomenal. We thought there that may have been due to mucus being produced by corals on the nearby reefs being swept out and sinking down through the water column near the reef where we were diving. But here we're off Fraser Island, so I don't believe there are any large reefs down this way, are there?
No, Dhugal, we're not really in the vicinity of any reefs where we are at the moment. We're quite out in open water.
So you wouldn't expect upwelling, and there's no reefs for lateral transport of mucus. That's quite a lot of particles, nevertheless. Siphonophore. This water is part of the East Australian current, I imagine, is it? Flowing south from further up north?
Yes, it definitely is, which we have to contend with.
Siphonophore. So the plankton that we'll be seeing here will have been swept south from further north. And as we go deeper, we'll enter a water mass where water from Antarctica, or the convergence zone around Antarctica, will be flowing north in the opposite direction. So we'll have a layer somewhere a bit deeper than this, where we'll have warm water flowing south and colder water flowing north. And the boundary of those two water masses is going to be a very interesting place with regards to the plankton. We should be able to see that through the salinity profiles in the CTD. The CTD is a conductivity temperature depth meter, which we have on the ROV, which is recording what the environmental parameters are as we dive. Mardi, what's the depth of the seafloor here? Do you know?
We're in about 3,400 metres here, Dhugal, so it's quite deep. And we're going to try and get down to at least around 1,200 metres for you today.
Excellent. So no chance of accidentally hitting the seafloor. That's good to know.
Yeah, I don't think we're going to bump into anything.
Be nice to bump into one of those big siphonophores. And I kind of only half say that in jest, because as you're doing these midwater surveys, bumping into the animals with the camera lens is a very good way to get good shots of the animal, because as we collect them as like you collect insects on the windscreen of your car. Somewhere as they approach the camera, they're going to be in focus, and we'll be able to get an in-focus image to allow us to identify the animals. So for midwater dives, we try and have the particles coming towards the camera as much as possible. So now, because we're slowly heading obliquely down, the camera is angled slightly down and looking forward in the direction of motion of the ROV. And we're trying to, we're trying to videotape the animals if we can, but if the water current is too fast and it's hard to actually stop and look on an animal, we try and collect them on the camera glass. Thank goodness, unlike insects on the windscreen, they don't stay there after you collect them. Ooh, that's something gelatinous. Just marine snow. And that looks like a siphonophore. And another. There must be food for those siphonophores here. Probably a lot of these little particles we're seeing are actually things called copepods, possibly ostracods as well, small crustaceans. Crustaceans like shrimp-like animals with hard exoskeletons. And a lot of these particles are probably going to be living animals rather than sinking dead detritus or marine snow. Very hard to tell, even with a 4K video camera, which of these particles are living and which are dead. If you look very closely at the particles and the way they're moving, sometimes you see some of them swimming to escape rather than just floating in the same direction as the other particles. And as we analyze this video back in the lab on shore, we'll be counting how many of those particles are jumping and how many are just floating past, as well as trying to identify the animals. This live stream we're doing now is in order to be able to stream it out to everybody live in real time throughout the world. It's quite compressed algorithm, which is used to reduce the file size, or the amount of data that has to be sent to reconstruct an image, which is great for seeing an animal and catching an animal to sample it later on and actually being there and taking part in stuff. But we're also recording in a much better lossless codec on the ship. So we'll be getting these massive video files. We'll be coming back to the lab, and we'll be analyzing the video at the original quality at which it's actually being collected at on board the Falkor. So although I can identify some of these animals, or I think I can identify some of the animals, many of the identifications and ratification of those identifications will be taking place back on land afterwards. So I may not be 100% correct on 100% of the IDs as we're doing this now, and I'll try and do a better job later on land.
You're pretty good, Dhugal.
I've developed a bit of an eye for identifying, it's another siphonophore, Fisonect, animals from out of focus or dark or jerky images through years of work using ROVs back from before the days of high definition TV even. So the large format televisions with the electron gun in them kind of time frame. And it was very hard to identify animals back then. A lot easier now with 4K video. You can often identify them by the way their bodies are angled or their escape responses or other cues that you only really get with a bit of experience. So sometimes I'll be yelling out the name of an animal, and it'll be like, what? How on earth can you tell that from that image? But if we can stay on it and get it in focus and take a good look at it, then we can say, oh yeah, it actually was that. Sometimes it's not, but a pretty good percentage of the time, those larger faraway shots or out of focus shots are actually good enough to identify the animals. And we're trying to work on here at JAMSTEC now a artificial intelligence machine learning system to be able to take those cues where, as far as a normal human observer would imagine, there's not enough information in the image to tell what the species is. But since I'm actually able to ID it, there must be the cues present there. So we're trying to work out what those cues are in order to develop a machine learning algorithm for identifying them. And this is very important data that we're collecting here with the SuBastian on the Falkor, courtesy of the Schmidt Ocean Institute, because we're getting information from an area for which there is no information in terms of images of the midwater environment. So all of this is a world first and very valuable for furthering that scientific research.
Dhugal, do you have data from a similar latitude in the northern hemisphere?
Yes, I do. I have data from Okinawa, the island south of Japan, from the Tropic of Cancer. So that's pretty close. Actually, I've got a few bits from further south of that and further north as well. So yes, we can do a northern hemisphere, southern hemisphere comparison of the communities. It's a very interesting thing to do. I also have data from off the west coast of Chile and out into the South Pacific Gyre that we collected 2 years ago. And so some data from over there would probably be interesting to compare with our data set from here on the western side of the Pacific as well.
And that's because the eastern and western might be connected through some of these currents and stratified water layers. Is that what you're thinking?
Yes, the direction of the currents in the surface layers, they will be opposite. So I think we're going to see some interesting stuff happening with the connectivity on the surface, and like, say, the animals that are used to having warm water here in the East Australian current. We're having all these warm water animals heading south. As they head south, they're carried into these colder water areas, and which ones of them are able to survive the colder waters, and which ones will die and just become food for other organisms. That was some very large tentacles that were out there. Yeah, that looks like a Forscalia Siphonophore, I think, the way it's spinning around. Maybe Forscalia asymmetrica. The cytosome was rather small. Forscalia formosa, which is another common Forscalia siphonophore, has a very long, elongated, almost Christmas tree shaped. You imagine a Christmas tree with the illumination lights wrapped around it. That's kind of what the cytosome of Forscalia formosa looks like. Asymmetrica is normally much weaker looking, I guess you could say. They're very interesting siphonophores. Most siphonophores have pairs of swimming bells, which are situated opposite each other, and they kind of open and close, forcing water out, and they're opposing. So the left and the right, opening and closing will move the animals forward. Forscalia, they're arranged in a spiral, so rather than swimming like this and going forward, they spin as they go. because they're arranged in a spiral. Sorry, we had a map up just then of the area that we're diving in off the east coast of Australia. Okay, so we can see there's a lot of deep water and then a very, very narrow channel. We're diving in the deep water mass. Ooh, is that a squid? What do we have here? Wow.
We're just going to try and focus in on
Oh,
that a bit for
it's
you.
it's a heteropod. It's a mollusk. A mollusk. The shell is pointing down. That slightly conical-looking shell. The bright white spot on the end of the trunk there is the buccal mass, or the mouth. In Japanese, this is called the elephant jelly. It's not actually a jelly. It's a shellfish, a mollusk. And to the right is its tail. Top left is its head. You should be able to see two black eyes with a nose like an elephant's nose hanging down in front of it. Oh, it's wiggling its nose around. And it has a single fin, which it's flapping now to swim, and flapping its tail. And then the heavy shell is heavy, so it's the thing that's pointing down. A lot of its organs are inside that shell there. That's nice. What's that, pterotrachea? Not sure of the taxon there, although I do know what page of my jellyfish book to look at to find it. Just can't recall offhand. That's very nice. Looks like it's got something in its gut as well. That's the great thing about transparent animals, is you can actually see what they've had for dinner. I believe these guys mostly eat other gelatinous animals, transparent animals. So when they do ingest them, they don't make themselves more visible by having something heavily pigmented inside their guts. It looks like this guy's eating something that's kind of opaque though. Oh, he's moving that fin around, trying to get out of here. Look at him go. And in order to swim, he's actually holding his I'll say nose, his trunk. The mouth's actually on the end of that, so it's not really his nose. He's holding that kind of backwards, so the area around the eyes where the head is the bit that's most forward in the water, in the direction of movement. Animals such as these pelagic mollusks have calcareous shells, and these shells can dissolve in the ocean waters as they become more acidic, as more carbon dioxide dissolves in them.
Dougal, is
So
this
there are
something that you might be interested in collecting, or are we just getting vision of this one and then move on?
This vision is fine, and then moving on. This is an animal that can be caught in plankton nets. So for people doing genetic studies, etc., they are able to get material to do those using regular sampling apparatus. We're here with the ROV, and we can collect animals that do not survive collection in a plankton net. So any jellyfish, ctenophore comb jellies or siphonophores, other jellyfish that we catch with the ROV here, the chances that anyone will have a specimen from off the east coast of Australia and that specimen is in good condition is very, very low. With the ROV, we can suck them up in what amounts to an underwater vacuum cleaner. Oh,
is a jelly?
Yes, and it's a ctenophore, I think. Could it be Thalassocalis? Does it have two separate lobes, or is it a Medusa kind of a shape, looking like a bowl? Looks like it's got two lobes, maybe. Let's see how we go.
Just seeing how we're going in this current, Dougal. Try and
Roger
get a
that.
good stable image for you.
It looks like Thalassocalis. Let me type that in so we can get the spelling. Thalassocalis. In Japanese, it's called the mask jellyfish. Mask as in, it looks like you're wearing a mask, and the gonadal tissue looks like two eyes when you look at it from above. Because it's kind of caught in the current now, it probably won't look much like a mask to anyone looking there. But you can see the bright white spot in the middle is the stomach, and the two white larger crescentic looking masses on either side of the stomach there are the gonads. Most ctenophores are hermaphroditic, which means they have both male and female sexual capability. Oh, nice. Yes, yes. Okay, yeah, that's a Thalassocalis. That's a species level identification there. Ooh, look at those gonads. They're very developed. So it's a fully mature animal. It's got its lobes closed. I think that's not because it's actually eaten something, but just because it's been swept around a bit. Oh, very nice. Yes, yes. So it's pursed. It's actually got like a medusoid bowl-like shape, and it's got a ring muscle around the edges of the bowl, which it's now pulled tight. Normally that would catch krill or other animals inside, so they can't escape. And then it hangs its tentacles down inside that hollow that it makes inside its body to snare the animals to eat them. So it's a side-on view here, looking at the two gonad strips coming towards us. And then that's another side-on view from another angle. So now we have views from both sides and from above. So that's very nice. Oh, yes. Thank you. Wow. Awesome work.
Dougal, we've got Cody piloting today and J-Rod co-piloting, operating the camera and the zoom.
Great job, guys. Awesome images. I can tell you've done this before. It really is very difficult to pilot ROVs to look at midwater animals because these animals are usually neutrally buoyant, which means any movement of water, then they're just going to follow that water movement. So if you come up on an animal and then you reverse back because you're getting too close, normally the thrusters will like send out a stream of water in the direction of the jellyfish and will blow the jellyfish to smithereens, or just blow it out of the way so you lose it. You can't find it again because they're very transparent. So as soon as they get too far away from the ROV, the lights will no longer light them up and you won't be able to see where they went. I've been on many surveys where we had a lot of trouble trying to zoom in on these animals and get good observations. Oh, whoa, top right, what was that? Very long, thin animal. Could have been a fish.
Yeah, we got a glimpse of it. We're just seeing if it's still around.
That Thalassocalis we were imaging just before, we saw some of them further north off the Great Barrier Reef. So it'll be interesting to see if they're in the same depth down here. I would imagine probably they are, since it's the same water mass just heading south. But we shall see. So if we look at like this same latitude on the east side of the Pacific Ocean, you'll be getting animals that are used to having living in cold areas will be coming into warm areas, and by looking at those kind of places, you'll be able to tell what effect global warming or the warming of the oceans are going to have on the animals that are more adapted to the cold. So it's very important to get information from all of these different water masses around the world so that we can model what the effect the changing environment is going to have on the midwater fauna. Because the midwater is the largest biome on the planet for multicellular life. Life on land is just a tiny little, almost two-dimensional when you think of it in the scale of the oceans, place where things live. The average depth of the ocean is 3,800 meters or so, and only 200 meters of that at the surface has light. And then the seafloor is another tiny little, almost two-dimensional volume down, covering most of the globe, of course, but it doesn't have the same volume of water as the midwater does. Oh, wow. It's another heteropod. Looks like the same species we just saw. You can see the shell is towards the top right, and the bottom left, that white spot is the buccal mass, the mouth, on the end of that trunk-like proboscis. You can see that one wing-like fin flapping around in the center of the animal, and that big diamond-shaped tail on the top right. And there's another one. I think they actually eat salps, which are pelagic sea squirts, usually. Although what the animals are eating down here is still quite a matter of conjecture, because when you catch them in plankton nets, they sometimes eat the things that are caught in the plankton net along with them. So they're having a munch as they come back to the surface. If they're still alive after colliding at high speeds with the mesh of the plankton net, then they tumble around inside the cod end along with all these other animals that are collected at the same time. The jellyfish get broken into pieces, and there's a tentacle here or there, not much else usually. Animals like this, they can find themselves pushed up against some other animal, and they're like, oh, I can't breathe, munch, 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 munch. And when you look in their stomach, you'll think that that's what they eat in situ, actually down here when they're alive in the ocean. But sometimes it's only what they're eating in the net. So being able to observe these animals while they're still alive, especially if they're doing something like eating or mating, for example, is really, really valuable for telling what they're doing actually, and not just trying to guess at. Ooh, that was a big siphonophore in the background there. Larvation. Ooh, what's that? Huh. Oh, look at that. The area behind the head is expanded, almost like a fin to help it be stable as it swims. Huh. That's the other thing, is sometimes the animals, when you catch them in a net, they've already like shrunk up and they're like, no, don't hurt me anymore. And they've like retracted all their body parts. And they don't really like to relax again when you put them in formalin to preserve them, to take them home and look at them under a microscope. So sometimes the actual shape of the animals, when you see them alive like this, can be different to what you imagine they were like looking at specimens. That's an amazing back region of the head there. Really expanded. It's easy to understand why they're called the elephant jelly in Japan. It really does look a bit like an elephant with that trunk and the two eyes there. Okay, cool. 383 meters depth. I would be expecting to see some krill somewhere around here.
It's about 14 degrees C at the moment, Dhugal.
14 degrees C, all right. We've got another cone jellyfish, perhaps? Or is it a Vogtia, a Siphonophore colony? Maybe a Vogtia? Let's see. Looks like it's got a lot of tentacles hanging out at the back of it there. Yep. So I'd say this is a hippopodid siphonophore. It's a Calycophoran siphonophore, which means it doesn't have a float. We look at the top of that transparent jelly-like mass, which is nectophores. The swimming bells are all put together like a Jenga block or a jigsaw puzzle, all interlocking with each other. And then at the very top of that, I cannot see a bright white reflective structure, which means it has no float, which means it's not a physonex siphonophore, it's a Calycophoran. It only has swimming bells, no float. Yep, the yellowish color of the tentacles definitely looks like a Volpia. So it's got all these interlocking, like horseshoe-shaped nectophores, which are all locked in together, and the tentacles come out from within that mass and are protected by the nectophores themselves. Normal siphonophores have things called bracts, which are like shield-shaped or leaf-shaped polypzoids, which have evolved to protect the animals from, they're like a defensive structure, which protect the stomachs and the gonads from attack by predators. The hippopodids, this group here, they've lost the bracts, and so all of their soft, squishy, easily damaged bits are pulled inside this hollow made between the nectophores there. So all that yellow mass there is the sexual organs, sexual zooids, the stomachs, and it's retracted all its tentacles in there now too, because it knows we want to take a bite out of it. What it thinks we do. So it's retracted everything and is, wow, it's trying to protect itself. It doesn't look to have those many like spines or like sharp projections, so it's probably Hippopodius rather than Volptia. I would say that's nice. Yeah, it's amazing how these separate nectophores, which really don't look very, the swimming bells don't look very streamlined when they're separate, but when they're interlocked together like that, they've got this beautiful curve on them that helps, you know, like the shape of a tuna or something, and it helps them swim through the water with the smallest amount of resistance. Thank you. Hippopodid siphonophore, probably Hippopodius. And that Latin name comes from the horseshoe shape of the nectophores. I think the Hippopodus, the pod is like foot, and the hippo is horse. Something else here on the downward looking calendar. Camera, that looks like Forscalia formosa. A little bit earlier in the dive, you might remember I said the cytosome is a lot longer and looks like the like a Christmas tree with all the lights on it. So that's what this probably is, Forscalia formosa. If it is spinning as it tries to escape, that's a good way to tell if it's a Forscalia or not. Yeah, very long cytosome that doesn't break readily when it's caught up in currents. Forscalia asymmetrica, for example, will break, its cytosome will break up pretty easily, even with a small disturbance. Formosa tends to hold on to its parts a lot better. And the nectosome is normally pointed, so that transparent looking jelly on the top there with that bright white spot, which is the float, the pneumatophore, is normally very pointy and conical, with the nectophores near the top being very, very small and then gradually increasing in size as they get closer to the tail or the cytosome. And each one of those bright orange spots you see there is a stomach, and each stomach has a tentacle. So this guy's got like 1, 2, 3, 4, 5, 6, say 30, 25 or 30 stomachs on him. Each one of those stomachs has a tentacle, so is able to prey on the food around it. So even though this animal is made up mostly of water and is pretty fragile, if you try to catch that with a plankton net, it would break up into lots of little pieces. It's got over 20 stomachs, which can each eat a separate animal. So the predation pressure exerted on the other midwater animals by this animal, which is mostly made of water, is actually quite high. Excellent. Oh, and this looks like a prayed siphonophore tail. Wonder if it's got the nectaphores still attached. Yes. Between each of those bright white spots, it looks very transparent and thin, so it's not a self-chain. I would say this is a prayed siphonophore cyphosome, and it's lost the swimming bells and nectaphores. So it's got no zooids there to swim it through the water. It may have run into a fish or something and jettisoned this long cyphosome, because the nectaphores can grow back new stomachs at the base of the nectasome. There's a budding zone where it buds off new stomachs and other zooids. And so the cyphosome here is just going to keep living like a ghost net, you know, in fishing when they lose the net, and the net will be floating in the water catching fish for days after it's been lost from a ship. The same kind of thing happens with these ghost tails of siphonophores, where they're shed by the colony when the colony is trying to escape, and the friction resistance through the water is just too great to pull this tail along with them, so they say bye-bye. And as they escape, these ghost tails continue to float in the ocean, catching food and living quite happily, not realizing that they don't have a head anymore. Nothing that's swimming them through the water, and they just float there eating and reproducing until finally they are unable to swim away from a predator and get eaten, or they get too heavy and the nectaphores, the swimming bells, can't swim them back up into a shallow depth, and they sink down, and as soon as they hit the seafloor, everything's going to jump on them to eat them. That was a prayed siphonophore tail, probably a rosacea or a desmophes or something like that, rather than the genus Prayer itself. The comidia, which are the stem units there, seem to be very rounded and were quite separate or far away from each other. So I'd say it probably was not Prayer, but it was a prayed. See if we can't find one of those with the nectaphore still attached, so we can get a species ID. That would be very nice. And that's one of the guys we'd like to suck up with the suction sampler if we can find a whole one. Very difficult to collect a whole animal like that in a plankton net, because even if you do get some of the parts, those guys can grow to 40 meters in length. So if you're towing like a 1 meter or 2 meter diameter plankton net through the water, the chance that you're going to catch all the pieces of a 40 meter long jellyfish is pretty low, well, impossible. Even with the smaller ones, you're likely to catch a little piece of the tail or like just the swimming bells. Oh, is that a Nakamedusa? Let's go in on him and see what that is. We may have a solmissus here. It'd be sweet if we do. We'll be able to tell if it's a Nakamedusa because the Nakamedusae have very thick, rigid tentacles which extend from the midpoint of the bell rather than from around the rim. And they've
J-Rod's
evolved.
just saying that when we spot something for you, they're just having to manoeuvre the ROV around a bit for you for the current. Just give us a little minute here and we'll adjust our position and then we'll try and get a good shot of that one for you.
Roger that. Oh, what are you? What are you? Wow, what are you? What is that? Oh, no, it's a phonoma barrel. This is, at least. Is this the same thing we just saw before? We must have been looking at it from above. Yes, so this is the discarded barrel. Looks like it was only halfway through making it, of a hybrid amphipod called Phronema, and there's a krill floating next to it. So it's caught a pyrosome, or a fire jelly, they're called. They're actually pelagic sea squirts, a colony of sea squirts living together, and it catches them and hollows them out, and lives inside its corpse. But it's obviously seen us coming and has swum away and decided not to finish the job and just left the barrel there. Oh, look at that donutting animal there. That's almost definitely a euphausid, the way it's doing those donuts through the water there. So there are krill in this layer. So we haven't seen any Solmissus then.
No, not so far.
I remember our first dive off the Cairns Seamount, they were everywhere at shallow depths like this, 400 meters or so.
Yeah, they were. I remember that. Yep.
Are we still at 14 degrees?
Just under 13.7.
Right.
110 meters.
Hmm. A lot of objects in the water, but they're quite small, so hard to tell what they are. Oh, that's another Siphonophore. That's another Forscalia formosa. And what's that beyond it? That looks like a ctenophore, perhaps. You concentrate on the guy on the top left.
Can you just repeat that, Dougal?
Not this Forscalia, but just to the top left of that, there's a ctenophore, I think.
Are you more interested in the ctenophore?
Yeah. I can ID the Forscalia from a distance and can tell what it is, but the ctenophores are a lot trickier. There he is. It's really hard because the ctenophores have very little contrast. They don't have nice
it's
bright.
really tiny.
Oh, okay. Yes, it is, isn't it? Wow. Actually, that might. Is that an oikoplurid? Is that a larvation? You see a little tadpole-like animal in the middle there with a bright white head? I can't see any comb rows. But it might just be my video. I can see something glittering. The color suggests it's a ctenophore. It looks like there were green reflections. Oh wow. What is that? It's going to be nice to look at on the good quality video later. I don't think I can ID it. It is small, isn't it?
Yeah, we're just about at the limit of where we can focus.
Is it a lobate? It's got two big lobes hanging out the end of it.
Yeah, I think it has, Dhugal.
Like it, yeah. It's a little lobate. It's not actively swimming, like flapping those lobes. So it's probably not a Bathysauroi, although some of those species don't actively flap the lobes. I wonder what it is. Oh, it looks like you're doing a good job of videotaping that. I should be able to tell what it is later. You don't fair. Right. Well, I think I'll just have to wait till I see the good video for that. It looks weird though.
Very weird and very small. I hope we managed to get something for you.
Huh. Something to look forward to. Going through these videos later in the lab, and you can imagine even just that short sequence there of several minutes is going to be like 30 minutes of going 1 frame forward, 1 frame back, 2 frames back, 3 frames forward, trying to reconstruct what its structure is to tell what it is.
And trying to
nice long hours.
Creative event logging.
Is that a discarded oikoplurid house? Look at that. Yeah, I remember my my first try at event logging was actually inside a manned submersible, looking out the window, and I wanted to be looking out the window the entire time, so I couldn't look down at a notebook and write notes because then my eyes would be on the notebook instead of on the window. So I was just yelling stuff out on the audio track so that I could write down later what I said during the dives. And man, there were some laugh moments when I went through that audio track afterwards. Here's another heteropod. Another one of those elephant, shellfish, jellyfish animals. Another heteropod.
You want us to get in an image this or keep on going?
Yeah, keep on going. I think we can tell what he is. You got some really good images of the previous one. Yeah, look at that expanded head. It's the same species we just saw before. Oh, they're cute. Wow. They make up a pretty big proportion of the animals we've seen, don't they? Like, as far as large animals go, we've seen 3 already, haven't we? Oh, thought I just saw a fish. And there's a donut, so that's a krill. When I say donut, I mean donut. swimming behavior, not that it looked like it was round and ring-like. If you notice at some stage the water temperature or the salinity rapidly decreasing, can you let me know, please?
Dougal Cody wants to know, what's your top 100 of species or organisms? We got time to rattle off 100?
My top 100 that, oh, wow, what's that?
That's 99.
Wow. My top 100 ones that I like, or top 100 as in how abundant they are? Wow, what do we have here?
Oh, this could be a solmicis.
It looks really small. Can you see tentacles?
Yeah, definitely. Tentacles. Lots and lots of particles in the water. There's a lot going on. I'll try and do a tentacle count.
Those look like a solmicis, isn't it? Or at least a cuminid. So after we videotape this, it would be great to catch him. It looks like it's eaten something. There's something in the stomach.
I think I'm counting about 13 tentacles, Dougal. Don't quote me on that just yet. Maybe 13, maybe 15.
Might be the guy I've been calling Gordon, for want of a better name, because it's undescribed. Look at all those krill. Man. Can you see the white spots that seem to be inside the bell, inside the stomach? I don't know if you're able to make out if they're like food, they're in the stomach pouch, or whether they're gonad material around the rims of the stomach pouches.
I'll try and get a good shot of that for you.
It looks like there's 3 or so I guess it'd be food. But 2 of them seem to be exactly opposite each other on the bell, which is weird.
So our team in the back here thinks it's 2 gonads and 1 piece of krill.
Mm-hmm.
So that's from Vicky, who's doing our vent logging today, Vicky Lowe. She's at the University of Queensland, and she's also joined in the back there by Luke Notiff from QUT. What would you say? Are you a biological oceanographer? Oh, sure. Yep, today. Today she's a biological oceanographer.
Awesome. Yep. If we can sample that, that would be very great. Are we able to sample this guy?
We're going to try and get stable enough to try and get that for you.
Fast current. Good luck.
It looks like there's 2 sitting here right now, Dougal.
Oh, really?
Yeah. One we have been looking at is the further away one, but another one has just come into the shot.
Oh, cool. Yeah, I want to catch the animal that we have the good video of, if possible. If we retake the video of this one, then that'll be fine as well.
So I think right now we're still looking at the original one, the one we were just looking at that we got video of. We're going to try and sample that now.
Sounds good. We're in the middle of all those krill.
Oh, the suspense. Looks like we got them. We're just going to check in the tube.
So small and transparent, it might be hard to see even in the canister.
Cody says he saw him go in, so we're pretty sure he's in the jar.
Awesome. Ente. Tagged and bagged. Bagged and tagged, so. Going in for a look to see if he's in there.
Yeah, we're just going to go in and have a quick look. We've got fresh new jars.
Oh yeah, they're very clear, aren't they?
Shiny.
Mm. I think our jelly might be clearer than the jars, though.
Yep, we see him. He's tiny.
Awesome.
Yep, see little tentacles in there. Okay, we're going to have fun with that in the lab this afternoon.
Great. Lots of vibrations on the ship as well, I'm sure.
Okay, we'll keep heading down.
Okay. Ooh, awesome. See if we can shake off these krill. They've all come to the light. think I've only seen a couple of fish so far. Another krill. Seem to be floating up.
Just give us 1 second.
You're getting the ship and the ROV in the right position, I guess.
Yeah, we just had a pilot changeover.
Okay. Yeah, it's intense concentration. Really hard to do that for a long time, I'm sure. Even, whoa, that was a jellyfish. Even one day
There
when
it
they
is.
get, oh, that's another.
Here we go.
How many tentacles does he have? It look the same as the last one?
They all look the same to me, Dougal.
Same with me and black corals.
It might have more tentacles, actually.
Hmm. It does seem, I don't know if it's larger or more robust or. Yeah, once we get a nice side on and a nice top down shot on it, it'd be great to collect him as well. If they are two different species living at the same depth.
Righty-ho.
As they grow, I'm pretty sure they probably increase their tentacle numbers by maybe going to double the number of tentacles as they kind of split.
Fish.
Oh! Oh, and a hybrid amphipod. It's all happening at the same time here.
You want us to focus on the jelly, right?
You want to do the fish, don't you?
No, we want to do what you want to do.
All right. Yes, I would like the jelly, please. The fish video seemed it would be good enough to get an ID anyway with what you had.
It's busy right
Nice
here.
hybrid. Yeah, you got krill everywhere. You got a sort of fatty skelet. Not sure what hybrid amphipod. And then our friend the Solmissus. And see, the krill are also, as well as the donut kind of swimming, they also spin around with their along their long axis. I think they can probably see polarized light, and they're using that. Man, they've all come to the lights. Is that a self chain? Salp's a good food for somnices. They go in.
Okay, we got him.
Excellent, thank you. We might need to do a bit of a hightail, like put some speed on the ROV to get rid of all of the krill that have swum in and seem to be following us.
Okay. Yep, we're going to do that.
Okay.
Okay, we're just going to start slowing down a bit now for you, Dougal.
Thank you.
Our CTD temperature logger looks like it's out at the moment, Dougal. Can't give you a temperature update just now.
Okay.
It's 12.5 degrees C at 450 meters.
Okay. That's a pretty rapid change, isn't it? You haven't noticed that a change in the direction of the current as you've been driving the ROV, have you? Aha, that looks like a siphonophore, maybe? Maybe not, no, it's a piece of mung. A discarded larvation house.
Dougal, the ADCP at the moment is showing that we're still in that current that's running from north to south. J-Rod thinks it might have slowed down a little bit.
Roger. Ctenophore? Lobate. Looks to be a lobate ctenophore. Oh, there's a cyclophone fish. I think I can see 2 tentacle bulbs. Yeah, it looked to be a lobate rather than a thalassocalus, unless the gonads aren't developed, of course. It'll have to be a job for when we get the good video later.
I'm still just trying to adjust our position and the camera focus just a little bit for you.
Great. Ooh. Volanopsid, maybe? Oh. The tentacle bulbs are on either side of the mouth opening, so it's not a Bathyceroe. It looks like a volanopsid of some kind. No black spots on the lobes, not that you'd expect infundibulum to be here. I'd say my inability to identify it to species is because it's either a new species or a described species that wasn't described very well when it was first described. But the video should be good for looking at what it is. It's a nice side-on shot there, yeah. Ooh. Is that a salt chain or a siphonophore? Looks like it might be a salt chain. Moves like a salt chain. Yeah, tastes like a salt chain, feels like a salt chain. I think that's a salt chain.
I was just saying that to myself in my head.
So it looks like the same structure repeating itself with no separate, like swimming part and feeding part. So it was all the same structure the whole way along the animal / colony. The round spots were the stomachs, and they were joined together in a chain. And the chain was not like salps next to each other or in a circle or a helix. They were kind of end on end. So it was probably the genus Salpa within the salps. And that looks like a discarded Bathycordius or larvation house. Oh, and what's this? Maybe a Siphonophore. And another Siphonophore. Fish. Maybe Calycophoran. Wow, what's that? Looks like a Physonex Siphonophore, the orange bit does anyway. What's that great big massive mucus? Oh, is that the tips of the tentacles? Is that in feeding position? Little Physonex Siphonophore with the nectosome to the right, the bright white float at the top there. It looks kind of coiled up in a retracted siphonosome, but there's this big cloud of something around it. Is it being caught in the cloud of maybe the cloud? Wow. That's weird. It's just sitting in front of it. Huh. Did it just start sinking? Or is it swimming actively down now? Looks like it's swimming, doesn't it?
Looks like it's on fire, that's what it looks like.
Yeah. That is really weird. I wonder if it's a Mars. Wow. Do you think you'd be able to collect this?
We can try. Do you want a bit more imagery first, Dhugal, and then we'll try and collect it, or do you want to just take the
If
opportunity
you're able
to try and grab
yeah.
it?
Yeah, I doubt we'll actually be able to see anything more that we haven't seen in the imagery, though. Unless you can get really tight. Oh yeah, nice. Yeah, okay. Proven wrong. Wow, that's... It might be one of those guys who will eject its tail when it gets... Oh, it's not holding together pretty well, isn't it? Ah. So what on earth is it? looks like a species of Maris that we get up here in the northern hemisphere, but it looks different again. I wonder if we might have a southern hemisphere congener. Yeah, let's grab him if we can.
Yep, we're
Very
getting the
important.
suction sampler in position now.
Right.
Got him.
Oh, is that all of him?
Yep.
Great.
Looks like most of him anyway.
Yeah,
We're calling this
awesome.
guy on the event logger, Dhugal.
Ooh, the fire siphonophore, the flame siphonophore. I think it's a Marus, M-A-R-R-U-S. It looks like a Marus orthicana, which we get up here in the northern hemisphere, but they're not supposed to be down there. But that doesn't mean they're not. Oh yeah, he's bagged. There's also another Marus species, Marus antarctica, which this could be if it's been swept north.
Do you want to look at it in the jar, Dhugal?
I saw it go in. No, that's fine.
Yeah, no, we saw it go in. We're good.
There's a Marus species which is very similar to Marus orthicana that occurs down near Antarctica. And it could be swept this far north in the Antarctic intermediate water, but the water mass we're in right now is still the one heading south, isn't it? So if it is that, it's actively swum up or floated up into the warmer water, which will give us an idea about how warm temperatures it can survive in, which is really cool. As the waters around Antarctica warm up, this will give us an idea. At least we'll have like how it can survive at that maximum temperature kind of data.
Dhugal, can you tell us a bit about the fiery reddy orange tail thing on that one? Do you know what that is, or about that?
Yeah, so it's the Siphonophore. It looks like you've got a phronema about to come into view as well. It's a pram bug. And this one's got the female amphipod still inside the barrel from the look of it. Yep, it's pulled itself in and is sitting in there in the barrel. None of its legs or head hanging out, being protected inside the corpse of a pyrosome that it's hollowed out and is now living inside. It'll lay its babies on the internal wall of that tube, and will go out and eat jellyfish and tear off little pieces of them and send them into their children to eat. Phronema sedentaria. So yeah, the red cyphosome, all of the zooids seem to have an orange color rather than just the stomachs, which is more usual. And Marus orthicana, at least, I know makes a lot of mucus, that orange kind of mucus, not sure why. And this species looked to be too, which is why I think it might be the southern hemisphere congener of Marus orthicana, Marus antarctica. But there's still debate as to whether that's a separate species as far as Siphonophore taxonomists are concerned. So this will give us DNA barcode to compare to the northern hemisphere population, for which I have several specimens, plus a physical specimen to look at the morphology on an animal that hasn't been almost crushed to death in a net. A very valuable sample, I think. This phronema has just pulled all its body parts in so it doesn't get hurt. They actually stand up pretty well to collection in a net because they hang around inside the barrels, and their body proper doesn't get squashed, only the barrel. Thank you.
Dougal, just wondering, we're at 465 metres now. Do you want us to pick up our descent a little bit just to make sure we get into that Antarctic intermediate water for
Yes,
you
please.
today? Yeah, okay.
Yeah, you bet. So what will be our next discovery?
Dougal, would we be expecting that Antarctic intermediate water around 800, 900 metres, something like that?
That's where it should be, yep.
It's all right, that gives us something to aim for.
You'll see the salinity rapidly decreasing when we start to enter it. Probably be several hundred meters in thickness. Not quite sure how much of it.
Descent rate right now,
Yep.
or should we speed up a little bit? Or still?
No, that's good.
This is good. Yep.
Yep. I don't want to miss the stuff that's right at the boundary of the Antarctic water and the northern warm water. This is just so great being able to see, like, dive into this water mass. I've been wanting to come here for 15 years now.
Oh, that's awesome. I'm so glad that we could do this for you.
Colobanema sericium. Oh, and Pitari right on 500 meters. That's where it lives up here in the Northern Hemisphere as well. How do you like that? Completely different environment and living at exactly the same depth.
Can you just say the
They
name
must
of
have
that
pressure
one again,
sensors
Dougal?
on them. Colobanema sericium. Colobonema. Colobanema sericium. Sericeum. Just rolls off the tongue.
And is this imaging and a sample, or just imaging?
Just imaging is fine. Yeah, the pulling in of the distal tips of the tentacles like this, and the very bright white tips, and the bluish color of the entire animal is very indicative of Colobanema. It's got that very long, narrow, thin stomach hanging down there from the top of the bell. These guys, when they escape, will drop their tentacles, and the tentacles will glow and act as a decoy, kind of like when a lizard drops its tail. And so the Colobanema will swim away while its glowing tentacles are there attracting the fish or whatever it was that went to try and eat it. So you can tell how many times a Colobanema has almost been eaten in the past by looking at comparing the number of long versus short tentacles, because after it throws the tentacles away, it grows them back again. So if you've got like tentacles of 3 different lengths, it means that it's been attacked at least twice. Nice. This guy seems to occur everywhere, at least from tropical to temperate waters. Not sure how far south it goes.
Happy for us to keep going?
If you get the right... Yes, thanks. Keep
Yep.
going.
degrees C,
Japanese,
Dougal?
11 degrees. Japanese, that's called the rainbow jelly, because when you shine light at it from the side, the muscles are so developed that it's bifringent. The muscles scatter color in lots of different colors, so it looks like a rainbow. Very nice to photograph in a photo tank on the ship. And a lot of the time the animal is intact when it comes up in a net. It's got very hard mesoglea compared to a lot of the midwater jellyfish. They never have their tentacles on them, though. Of course, they jettison them as soon as the net comes in contact. I actually met a researcher once 20 years ago when I first started doing this stuff who didn't believe that Colobonema actually had tentacles. He thought they'd like evolved to live in the deep sea without having tentacles. But that's just because all the samples they'd seen had been caught by a net. Just shows the value of, oh, was that a Tuscaroid radiolarian maybe?
So it just so happens that we have a radiolarian expert in the room with us at the moment, Dougal.
Oh, okay.
Kilo.
Did they get a good look at
with
it?
us.
It was on the...
Oh, it was awfully quick. We're gone now.
They're amazing creatures, those Tuscaroids. Tuscaroid, are they? Maybe a radiolarian expert can tell us a little bit about them while we zoom in on this. Is it a larvation house?
She deals in Southern Ocean radiolaria. She doesn't want to talk about tropical waters. So Vicky's doing her PhD at the moment at the University of Queensland. And I think she's really a paleoceanographer. Is that fair to say, Vicky? Yeah, she's a paleoceanographer and uses radiolaria purpose.
Microfossil.
Yes.
Ooh, is that another one of those Bolenopsids that we saw just before? Be nice to get some good video of this, because more than one observation of the same species lets us know a little bit about the inter-individual, intraspecific differences in morphology. So we can actually say, yes, it is new, or no, it's not new, rather than, it could have just been a mutant, we're not sure. Those lobes are so compact and small. So it's holding the lobes together, and like in a textbook, you often see these ctenophores drawn with the lobes and the mouth down, and the aboral organ pointing up on the page. Almost every textbook in the world has ctenophores drawn like that, but they're all drawn upside down, because usually the mouth is pointing up, or it's going forward with the mouth pointing forward in the direction of travel. So yeah, it'd be like, you know, when I open a textbook that's got a picture of a ctenophore in, it'd be like an ornithologist studying birds who opens up the textbook and all the pictures of birds are upside down on the page. But because nobody has really seen them in situ, a lot of these species actually alive underwater, it's easy to see how you can be mistaken about you know, which way is the right way up.
Time to rewrite the textbooks, Dougal.
Yes. It reminds me of the story of a squid called a cockeyed squid, where they've got one big eye and one small eye, and a researcher had caught one of these squid and put it in a fish tank and was filming its behavior, and noticed that the large eye was pointing down and the small eye was pointing up, and hypothesized that the large eye was big to collect all of the faint light as it was looking down, and the smaller eye was smaller because there's more light if you're looking up towards the sun. And all this was hypothesized, but we actually saw the animal in situ during an ROV dive. They sit vertical in the water. They do have one eye pointing up and one eye pointing down.
Here, Dhugal.
It was basically that the squid was half dead and the heavy eye was pointing down. Yes. A ctenophore, it looks like.
No, we're saying not ctenophore.
Still in one piece. Oh, it's a Narcomedusa.
Yeah, I think so.
I can see tentacles now. You get a tentacle count on that?
Do you want to sample this, Dhugal?
Yes, I would.
Yes, please. Okay.
Yes, please.
Just bear with us. We're also kind of manoeuvring relative to the ship at the moment as well.
Right. Does it look like it might be the same as the last species? I can't see any obvious differences. I didn't really see it that well. Wow. Okay, that's quick. Awesome. Thank you very much. Okay, let's keep going down. Looks like there might be fewer krill than there were about 100 meters shallower. It might just be because we are moving more and they are not attracted to the light, so they might really not be there. Now that we're going forward, I can't see krill all over the place, but I think there are less. Siphonophore. We're lucky we might see some of those Antarctic radiolarians in the deeper layers as we head into the intermediate water. See what they look like when they're alive.
Vicky says that would be nice. Thanks very much.
Yeah. They have the most beautiful skeletons, don't they, those radiolarians? I don't know if you call them skeletons or tests or what they're called.
I think they're a test. Are they, Vicky? Yep, a test. Yep.
I failed my test. I call them skeletons. Test.
So we're just going to continue our descent right down, get down a bit deeper for you, Dhugal.
Thank you.
Just wondering how you got into this in the first place. How did your journey to jellies? And, oh, squid.
Oh, the squid. Oh, more than one.
Everywhere.
Oh, is it a squirrel? Oh, yeah. What are they, Almostrephids? Almostrephed squid. Oh, 560 meters. Cool. So these are the guys, the marlin and other things dive down into the ocean depths to eat during the day. All these guys who are hanging down deep probably migrate to shallower waters at night. Ooh, wow.
Wow. That's so cool.
Oh, he's glowing. Look at that. Look at the pulse. Wow. They're communicating with each other, or they just don't like the ROV? Oh, look at that. Oh, wow. That is so cool. I'm afraid I can't ID squid. Yeah, so how I got into jellyfish in the first place was when I was at university, I was studying fish and shrimps and what they eat. And we were catching things in plankton nets and looking at plankton all the time. And I didn't really look at the jellies very much. Something interesting coming up here, Siphonophore, it looks like. Could it be a Bergmania? Certainly has a nice long cyphosome. And if the nectophores have no circular structure or T-shaped structure in them, and they're just like bags, then it would probably be a Bergmania. I think that looks to be good enough video to see that. Looks like it's, well, looks like it has structure inside the nectosome, the nectophores, the nectar sacs. The colony looks like a Bergmania, but I have to look at the high quality video later to show what that is. The Physonect Siphonophore at least. Either a Bergmania or an algalmatid. If it's a Bergmania, it's maybe Amorina, definitely not elongata.
Shall we keep going? Or you want to focus on this for a bit?
I think that'll probably be good enough images to see it. Yeah, that's okay. Yeah, I think it is a Bergmania. Okay, thank you. Yeah, that's good. looked like it had patches of nematocysts on either side of the ostia of the nectophores, probably for defense, the things that are trying to chomp it from the side. Yeah, so I was doing fish and shrimps and things pulled up by the plankton net, and there were only very few jellyfish in the samples. And then when I entered JAMSTEC as a postdoc, I did my first dive in the Shinkai submersible, looking out the window, and almost everything I saw was a jellyfish. And I realized that it was because the nets had just been destroying all the jellies, and that was when I decided I needed to become a jellyfish expert. Just because there weren't so many of them around. Monterey and Woods Hole, I think, were pretty much the only places where people had been working with submersibles to do work on midwater animals. Oh, the Johnson Sea Link also, which is now retired from Harbour Branch in Florida. So 3 places really in the world. And Japan was just starting up to look at midwater systems using the submersibles and ROVs. So it was basically...
Might have a ctenophore here.
Oh, yes.
A few ctenophores.
That's a Bathysauroi. Look at that swimming, the frog kick. Which one? It's got a very small head and short comb rows. It's probably Bathysauroi fosteri. Very, very long stomach and larger head. It may be Longigalula. It's looking fosteri-like. You see whether the stomach is like black or red rather than white or clear?
Red, by the look of it.
Red, yeah. So probably Bathysauroi fosteri. And a Physonex, and a krill. There looks to be some good shots there. Okay. Let's keep going down. Thank you. Had a really good selection of animals today. A lot of different species. Most of the major groups. Kind of surprised that we haven't seen a parasome, given that we've seen 2 Phronema. Got to make those barrels out of something.
Tell us about pyrosomes, Dhugal.
Pyrosomes are pelagic colonial sea squirts. And they normally take the structure of a tube, which is open at one end and closed at the other. And all the little sea squirts, their siphons are pointing outwards outside the tube and sucking in water, and then they expel the water on the inside of the tube. So all of these little siphons or mouths, I guess you could say, pushing the water into the tube, which is closed at one end, and that allows the tube to move forward in the opposite direction to where the opening is. And so with jellyfish, they've got to like open and close to swim. It looks like scalp chain. So you can see them like opening and closing, swimming. Pyrethrum looks like that's an atolla, I think. Yep. Which one? Van Hoffenheim? You see the shape of that dark stomach from above, we'll be able to tell which species.
You want to see that
if
from
it's
above?
a cross shape or it's a cross or a clover. Oh, yeah, it's an atolla. It's not dragging. Is that dragging a single tentacle? I can't make it out on my video, but
it does seem to be. Yeah, that is what it looks like.
it is. But that's how you can tell if it's an atolla, if it's dragging a single tentacle behind it. And then the species you get, there's one species with a cross-shaped stomach. The others are usually more club-shaped or clover-shaped. And then the other characters you need to look at is the number of tentacles and the shape of the gonads. Good. And then if you have a microscope, you look at the radial septa, if they cross the muscle bands or not, but you can't really see that in videos. Does the bell look mostly transparent and only the stomach reddish?
It does look like that, yes.
Maybe then Hoffenheim. Or maybe a young one, ideally. Looks like we might have some game shots on there somewhere.
Is this a sampling target, Dougal, or just images?
No, just images, thanks. These are fairly hard and can be caught in plankton nets as well. He sure can swim, can't he?
Yeah, he's making me go cross-eyed. I don't know how the pilots are feeling right now.
Thank you. You got the ROV moving in three-dimensional space, the jellyfish moving in three-dimensional space. The people piloting the ROV with the ship is moving in a different way, and so you've got three different things moving in different directions. Yeah. Okie dokie.
It's like a big long
Thank you.
siphonophore coming into the vision now.
Oh, yes. It does look very long, doesn't it? I wonder which end is the head or the nectosome. Looks very thin, so maybe a physonect rather than a Calycophoran. The chlamydia are very spaced out. Wonder if it's still going? Oh yeah, look at it. Wow. Oh. That's the end, so that must be the back end. Looks like it might be a ghost tail. It looks like it's maybe hasn't eaten so much and everything's degrown. Yeah. Yeah, I couldn't see a nectosome. It was one of those discarded siphosomes, those tails of a siphonophore. What's that, squid ink?
We think so. We're just going in to take a little closer look. Not sure, Dougal. Gerard thinks not squid ink, but
Okay.
I don't know what it is.
Can't say I know what that is either. I'll say what it looks like, but I won't say it if we're streaming live.
We're certainly live. It looks like it's really holding its shape, like it's not dissipating like you might imagine with squid ink.
Well, some squids shoot pseudomorphs out of their squid ink that look to be the same shape as the squid, and they use them as decoys. And the ink is actually caught up inside a mucus because they have a mucus gland as well. And so the ink tends to keep its shape very well. So it could be a like an ejection, one of these pretend squids made of squid ink, a pseudomorph. But then again, it could easily be something else. It's definitely good food for whatever is going to eat it as it sinks down through the water column. It could be a fecal pellet from something very big as well.
That's what J-Rod said, not quoting those words though.
Oh. Oh. That's a fishing siphonophore. Wow. It's all spiraled up. What on earth? So it looks like the nectosome is pointing up and the tail of the siphonosome is curled around in a C shape. And maybe one of the stomachs is really, really full. Is there like a big brownish stomach bulging there in the middle?
Yeah, it does look like that.
Oh, and it's sensed us, so it's pulling all its tentacles in. Look at that. Whoa. It's getting ready for its high-speed escape. Which probably won't be that high speed. Could be an apolemia, maybe? If it has tentacles coming out of the stem in between each of the nectophores, it'll be an apolemia. Doesn't look quite fuzzy enough to be one though, so it doesn't look like there are masses of palpons on the cyphosome, so I think the jury's out for that one. Does look like it's eaten recently though, more than one stomach. And there's a krill. Very fragile animals. We had probably better keep heading down. Nice. There's definitely a lot of animals here. Is this area a productive fishing zone? I should know that since I come from Rockhampton, which is fairly close to where you are now, right?
I'm thinking the same thing, too. Like, I should know this, but I really don't know. I'm sure someone does. I wonder if anyone on the YouTube chat is talking to us about that at the moment. I can't see the chat where I'm sitting.
Is this radial areas? Is this two radial areas joined to each other, or is it an oikoplurid house? Ah, it's an oikoplurid house. Sorry. I just got someone excited, I'm sure. It's a lavation house. You can see the inner filter and the outer filter both. You'll have to throw a fishing rod over the side to find out.
You're not the only one thinking that right now, Dhugal.
In the interest of science. String. Yeah, so the first time I ever did a dive in a sub, looking out the window and having to like have my lab notes or my observation notes as me talking on the tape at the time. Oh, there's another Colobanema sericium. Quite deep, this one. That's fine, we don't need to stop on him. I can ID him. Thank you.
Dougal, I do have.
But I had to give names to everything, even though I didn't know.
Right, yeah,
What's that?
There's a question on the YouTube comment feed at the just now asking what's today's mission and what's getting collected and what are we looking for. Do you want to just go over that?
Yes, so we're diving in an area now where I would expect the effective Antarctic intermediate water will be the strongest of any of the dives that we've been doing here off the east coast during the upcoming cruise and also the previous cruise we're doing over the summer or the winter over there in Australia, summer here in Japan where I am. And we're very interested in looking at the biogeography of the animals, why they're living where they are and where we can expect them to be living in other areas, and how that relates to the physical environment they live in. So, for example, there's a jellyfish called the red paper lantern jelly, Pandea rubra, which has been caught off Antarctica and is caught in the North Pacific as well. And looking at the genes, it seems that the animals in Japan are a subpopulation of the ones from off Antarctica, which means even though conventional wisdom doesn't say that midwater water masses can make it from off Antarctica all the way off Japan, we're getting jellyfish that seem to have been transported there because they've got the same genes as the guys who live down here. But there is not a single record of the animal in between Japan and Antarctica. So even if we see one during these dives here, we can say they do occur there and they live in this particular depth and this particular water mass to try and model where else we would expect to find them, and then hypothesize how they're actually able to be transported from off Antarctica all the way to Japan. And that'll give us a look into oceanography as well as just biology. Looks like a siphonophore there, Phytonex siphonophore. And one of the groups that we're particularly looking at is the dinner plate jellies, or solnices, where it looks like there are multiple species, cryptic species, which are hidden within one species name that people use now. And so we're trying to collect these Narcomedusae during these missions so we can get DNA barcodes and good morphology, as well as good distribution data on them. And we'll be able to try and piece together then which species live where, do they have different niches or not, and how that relates to biodiversity maintenance, because they're very similar morphologically, but if they're different species, how can they coexist? Are they eating different things? Do they actually like different environments? Are some of them thriving in a place where others have just been passively transported there and they're on their way to die and they're not feeling so good? So these kinds of questions. And especially with regard to the Narcomedusae, they're very voracious predators. They eat anything else that's gelatinous in the mid-waters and apply top-down predation pressure, stopping the jellies from overtaking the world, if you want to look at it that way. And so what happens to those animals as the oceans warm, or as the oceans become more acidic, or as the ocean oxygen concentration goes down, is something very important to understand. So if we know what the maximum temperatures they can take, or lowest oxygen they can live in, we can model what will happen to the oceans as the environment changes. So getting information such as this from different places around the globe is very important, because you only find extremes of each of these different environmental parameters in different oceans. The waters off the east coast of Australia, for example, are fairly low pH acidic waters, so it's a good data point for looking at global ocean acidification. If you wanted to look at the effects of temperature, you go to the Mediterranean, where it's very warm, even though it's deep, and things like that. Very important data points that we're putting on the map here during the present expedition, thanks to the Schmidt Ocean Institute and our team on board the Falkor there, and flying the SuBastian, the ROV, because none of this would be possible, especially now with coronavirus messing everything up everywhere. We have our team there on the ship that's been at sea for goodness knows how long, how many times, and doing the work of multiple scientists all by themselves for those of us who can't be there. So big thank you for them, to them. We had some more squid just then.
Yeah, a bit of a squid party. They've been following us for a little bit now.
Yeah, and the fishing must be great there. No way you'll catch one of those squid. I tried to suck one up in a slope gun once, and it used its arms to like hold onto the slope gun, saying, "I'm not going into that tube." We had to give up in the end. Just couldn't suck it through. Wow. You think about it, it's quite deep. Like, they're very meaty animals. They must take a lot of energy to survive swimming around, actively hunting their prey and swimming away from predators. So they must be moving a large vertical distance in the water column to get up to where all the food is. Ooh, what do we got here? We finally have a medusoid-looking medusa. What is it? Baby piranha? Or is it a hydra medusa?
Just bear with us for 1 second, Dougal.
Yeah. Does it have radial canals or not? Oh, it's a very lumpy mesoglia. And large, wide stomach and a little frilly mouth on the end of it, maybe? You see if the tentacles are leaving the rim of the bell or whether it's got tentacles leaving partway up. I can't even tell if it's an Archimedes yet. Doesn't look to be. Oh, it's got lots of radial canals. So it's an Aquioria, or an Aquioridae maybe.
Is that what you mean by tentacles on the rim of the bell?
Yes, you can see all the radial canals with the gonads, the bright white gonadal tissue running along the radial canals that look like stripes coming out from the center. And then a fairly small number of tentacle bulbs on the rim and a very large, wide stomach with a round, like a rounded stomach with the shut closed there, and like the lips are like tassels hanging down from the middle, and then a great big lump of jelly or mesoglea, which is probably being used for flotation. They can change the concentrations of ions inside the jelly to make themselves lighter or less dense than the surrounding seawater. And nice. No, there's an Aquioria with a small number of tentacles but lots of radial canals, which I think is found off Australia, so it might be that. I can't remember the species name right now. It looks like a radiolarian with a string of mucus on it. Okay, thank you.
We can keep going.
Yeah, keep going down.
Keep going.
I'm going to get right into the Antarctic water. Nice one. Wow, what's that? Something sinking,
Thing.
maybe.
Yeah, I think so.
Fleck of paint, maybe. Yeah, it's not something alive. Coming up on 850 meters. Are we down to like 8 degrees or something by now?
We're down to 6.2.
Oh, wow.
Yeah. Getting there.
Is there a salinity readout there as well?
.43 is salinity. 6.1 degrees C.
Okay. 3, nice. Decreasing salinity, decreasing temperature. What are we going to see? Oh, shrimp.
Shrimp.
Huh, that's the first shrimp we've seen. We're at 870 meters and it's our first shrimp. And I didn't even realize that was weird until seeing that. I got my eyes on the jellies. It's like, whoa. Got its antennae out on either side there. So probably is suggested. I think someone recently, maybe not that recent now, decided that all the things we used to call third year and suggesties needed to be split up into 20 or 30 different genera, and I don't know what to call them anymore. Full red suggested group. We've definitely got less biomass than we had a bit shallower up. Still a few particles though. Hello, hello. Green snow. No, we haven't seen this entire, this dive all last cruise was Lapictace, the red lobate. I don't think I remember seeing a single one. That's a discarded larvation house, possibly a Bathycordius outer filter. It's become clogged. Lots of large particles attached to it, and the animal that lives inside has discarded it and made a new one. So it'll be happier up in layers closer to the surface now, probably with its new house, filtering food out of the water. And this clogged house has been thrown away and is now sinking down slowly to the sea floor. Not that slowly, actually, because they can sink up to 1,000 meters a day. Ooh, what is this? Is that a copepod? An amphipod. Is that a really big copepod?
Yeah, I think so.
Yeah, like a megacalynus. It's big. Has it got something clutched in its legs? It looks like it's got a white patch. Wow, it's huge.
We're having a discussion here about what's big or huge. These are relative terms.
I wonder if it's grabbed a piece of marine snow and it's chomping on it. Yeah, big for a copepod, right? So it's like all of several millimeters. Could it be a centimeter? You reckon it's that big?
Yeah, J-Rod's nodding. Yep.
Yeah. Godzilla calynus, we'll call it.
Do you want it?
Okay,
or
let's
looking
keep
at
heading
it?
down.
Right, keep heading down.
Just looking at it. Keep heading out. I have to keep forcing myself to put a virtual blindfold on because I'd love to just hang around and look at this stuff all day, every day, but time is too limited. Oh, was that a ctenophore?
Yeah, Chris says ctenophore.
Oh my goodness. No, that's a... What on earth? Is that a ctenophore? Oh my
A
goodness.
few heads nodding in here. Money's on ctenophore.
Holy mackerel. That is weird. So it's loosely a lobate, I guess. What on earth? We can, yeah, get this from as many angles as possible. That would be awesome. This is weird.
Yeah, we're going to maneuver around and see if we can get some shots from different angles.
Has it got bits of it that are yellowish and bits of it that are bluish as well?
Yes, correct. That is what it looks like.
Wow. Trying to get my head around the 3D structure of it, but this is... I can't even find points of reference. See the cone plates. Oh, we're looking at it from below.
We're looking at it from above at the moment, Dougal.
Are we? Does it look like it may have been damaged and is growing back, or is this it?
That's what J-Rod is thinking too. Yeah, it might be damaged.
Right. And it's growing back in some kind of a funny way. I still don't know what it might be though. And it's very big, isn't it?
We've just put the lasers on. Can you see that, Dougal? Just quite see. Oh, can you see the lasers there?
Oh, just a one-headed.
10
Yeah.
centimeters.
The animal's more than 10 centimeters?
Yeah.
That yellowish looking thing in the middle, is that its stomach? Or is that something that's parasitizing it? It must be damaged. Doesn't look like all the bits are there. Oh, that's nice. All right, so that's one lobe. They're the two lobes, okay. For such large lobes, there's hardly any canals passing through them. Yeah, I think maybe we've seen enough to know that this is not how it would look if it was normal.
Okay. All right, so we'll
we
keep
should
going.
keep going and try and find one that's
shooting
intact.
down a bit deeper.
Yeah, if we can find an intact one, that's a really weird animal. It just shows you how they can survive extensive damage. Because it was still beating those comb rows, like it was fine. It probably regrow itself over the course of weeks or months.
Here's a jelly. Here's a jelly.
That's a Solmissus again?
I think so. Like it.
Ooh! Is this one with more than 30 tentacles? 1, 2, 3, 4. Mr. Mike?
I'm not sure if it's more than 30. By 20s.
This could be the morph from that we caught near Antarctica. So if we're able to sample this after we video it, that would be really good. Oh, it's got gonad tissue. Look at that. It's on the edges of the stomach pouches. There's these bright white patches. They're actually brooding pouches where the larvae develop. They become plannular inside those pouches, and we don't yet know whether the planula are released into the water and develop into little jellyfish there, or whether they grow into little jellyfish while they're still in those pouches and live on the adult for a while, for example. We still don't know that much about them, but these are well developed. And there's only a pair to each pouch. Oh, there's one that's got 3. So it's either still young or it's just a morphotype we don't have the specimen of.
We're at 900 meters, Dhugal, and it's 5.7 degrees C.
Okay. It looks slightly purplish.
Sorry, did you say does it look slightly purplish?
Yeah. Is it clear?
Maybe at a stretch.
At a stretch. Okay, so not too clear. It's going to be too big to fit in the tube.
We can try and put it into a bio box if you really want it.
Really close.
Yeah.
I think the images are good enough that as long as we get a pizza slice, we'll be okay.
Okay, so we'll try and suction it, and if it breaks, it doesn't matter.
Yep. Oh, oh, almost. Oh, yes.
We got it.
You have to make sure that goes into the canister. It'll be stuck in the tube somewhere, probably. Oh, no, there it went. Oh, yeah, it's in.
And there's a squid party happening right behind us. In front of us.
Very nice. Oh, squid party in front of us as well. It's amazing how they change color like that at a depth where there's no light. Why? Maybe they just dilate those chromophores so that the bioluminescent light can't penetrate it anymore. Thank you. Yes, keep living Solmissus. The elevation house. It's like a jelly off there in the distance. Siphonophore? Or a house? Looks house-ish. Yeah, it's a discarded larvation house, a big one. That's a big food packet. it's probably all sorts of like egg masses embedded in jelly and mostly transparent. And some of these things we think are marine snow could be all matter of different things.
Dougal, getting back to that conversation about the top 100, Eddie would like to know what's your favourite subsea animal? Top one, number one favourite.
Number one favourite subsea animal. I think it'd have to. It's not actually a jellyfish, I would say. It's an anglerfish. One of those yeah, the ones with the bioluminescent frilly lures below their chin, as well as a lure on their head as well. And those big female anglerfish with, I think, a lure that hangs off their chin is light that's produced by the anglerfish itself, whereas the one in the lure that hangs off its head is actually produced by bacteria that have co-evolved and produce light in that lure. They are just like really awesome, and the skin is so black it absorbs light. They've got all these like sensory organs scattered around the head to feel for vibration. So as soon as something comes close, they just grab it to eat it. And these anglerfish, I think what surprised me most when I actually saw real ones rather than just like photos in a photo book somewhere was they're actually quite small, most of them. So you think that you'd be looking at a 30 centimeter kind of animal, but in fact you're looking at something that's 3 centimeters long. That was a bit of a shock. But some of the anglerfish can grow quite large. I think the largest ones are like 1.8 meters. This big female anglerfish, they actually swim upside down, and the barbel hanging on the top of its head is actually hanging down towards the seafloor. And I think it's probably eating benthic animals that jump up off the seafloor to eat what they think is good food, and they end up getting snapped up by this big anglerfish there. So, yeah, they're really exciting. Jellyfish, there's a huge number of species, and they're all very simple. Oh, that's a hydromedusa, is that? I think. No, maybe it's just snow. Just snow. The jellyfish, what interests me is that even though they're so simple and they don't have like large-scale migrations or like bird song or like mating dances or anything that you normally think is necessary to have high diversity, there's still a whole lot of different species, and they're all coexisting. And I just can't wrap my head around how so many species of jellyfish can coexist in an environment which is basically just a big mass of water. So there's no trees to hide behind or under the bark of, or digging under the ground, or any of these other things that heighten the biodiversity in places like tropical rainforests. You just don't get out here in the pelagic. So how on earth can so many different jellyfish species coexist with each other? So I think rather than a specific jellyfish species being my favorite, it's the fact that there are just so many of them and so many different shapes and types, different solutions to the same basic problem of living in a low food, desert-like and deep environment. So many different ways to come up with a solution without having this really high-level behavioral diversity is what's really cool. But if I had to pick a jellyfish, it would be Pangaea rubra, the red paper lantern jelly. Japanese pubs hang red paper lanterns outside them, and this jellyfish like folds up just like a red paper lantern. Oh wow, what's that? To the top of the screen, not the jelly, the fishy thing. Long white, yes. Oh, don't tell me that's just a string. It'll just be a string.
It just broke off.
Maybe it's just a string. I thought for a minute there it was an interesting fish. Yeah. Yeah, but the red paper lantern jelly, it has anthropods that live on it. It's got sea spiders that live on it. It's got other jellyfish species that live inside it. So it's a place where lots of different animals are dependent on it as a substrate. Just like a tree in a tropical rainforest can have an insect species living on the leaves, another one on the bark, another one on the roots, kind of thing. Well, jellyfish provide a substrate for other plankton to live on or in as well. So the high diversity of jellies is actually increasing the diversity of the other animals that are living down there as well. Another interesting thing about having so many species of jellies is, and why there are so many, there's a possibility that they all have different kinds of venom because they catch their prey using stinging cells, nematocysts, and they have multiple kinds of venom inside these nematocysts. And if each of these jelly species, of which there are thousands, oh, there's another squid. Couple, yep. If they all had a different kind of venom, then that's a lot of different neurotoxins that could be possible medicines. Oh yeah, there's squid everywhere, 960 meters. That's so deep.
We can see them all popping up on the sonar ahead of us, Dhugal.
Keep your eye out for a really big fish that comes down to feed on them too. That would be cool. Have like a marlin or something come swinging in and grabbing a squid. I've had a very large squid come in and feed on a squid like this when we've had the cameras on. It was pretty amazing. They like to eat each other, some of those squid. Jellies eating jellies, squid eating squid. Ketognates eat other ketognates quite commonly. Anything that might be food gets eaten. Strange how they just kind of turn up and then they just disappear again. Where all the jellies go? Be down to 5 degrees or so by now?
Yep, 5.4.
5.4.
975 meters. Oh, there's the squid. Here they are. This is amazing, quite something. Whoa, what?
Siphonophore? Just a ghost tail. Makes it hard to spot jellies when there are so many squid around.
There's a lot going on.
Imagine if you're a jelly and you're 98% water and one of those squids swim through you, there wouldn't be much left. You'd end up like that ctenophore we just saw.
And then it looks like they're gone again.
Yeah.
Just hit 1,000 meters.
Right, so
Probably got
you'll
about
have to get
40,
a
45 minutes left to go, Dougal.
Roger that. Oh, it's a classified. Which one? We need to see the somatocyst in the upper nectophore. This is like a rocket ship siphonophore, they're called.
Okay.
And does it look to have ridges, I think? Rather than being a rounded cone, angular. I think it's coniferous. And if we can see the somatocyst, if it's elongated perpendicular to the main axis of the siphonophore, it'll be multidentata. If it's not, it'll be Mosera. It doesn't look like multidentata. Jellyfish. Named after Charles Kuhn, C-H-U-N, I-P-H-Y-E-S. In either case, this is one where I have DNA barcodes for Japan and Antarctica, but none in between.
Does that mean you'd like us to try and sample this?
If we could, great.
Okay, we're going to try.
When it senses you coming, it'll probably give one big squeeze and like, whoa, rocket ship away. I think he went in, didn't he?
We can still see him in one of the other cameras, Dougal. We're just trying to get him.
Okay. Yeah, these guys can rapidly change their swimming direction. They're real pain to catch.
Yeah, I think we got him.
Yep, keeping my eye on that canister there. Oh, something went in, I think. Yep. Yep, you got him. Way to go.
Yes, we got him.
Thank you. So when you were saying 40 minutes just before, were you meaning to be back on the surface? Or,
No, till we leave in the dive and start heading up.
Awesome. So we have a good chance to make 1,200 if we push it.
If we get more Dhugal, if they're small, we've got 1 jar left for you. If it's something big, we can probably, but if it's small, we've just got 1 jar.
I'm very happy with what we have, I think. Unless we find a second individual of that Solmissus, so I've got n equals 2 instead of n equals 1. I'll try and control myself on the sampling, hoping we find that second one. We'll have everybody break into a chorus of I want it all, I want it all, I want it all, and I want it now.
I think we're all a bit like that, Dhugal. You're not the only one.
Have to keep a canister for the first planktonic octocoral that we come across. Kind of surprising we haven't seen any coelodendrid colonies at all. I would have been expecting to see a few because they're so large. They should be in fair numbers.
What is that, Dhugal? Can you describe that to us?
So they're a single-celled animal with a ciliaceous, silicon-based test, and they can often have. They normally have like spherical main bodies and then long glass arms that kind of come out from the bodies. So they really stand out with all the stringy or round particles of marine snow that are down here. And then suddenly you see something that looks like a ball with 4 arms. And they're very bright white because the glass really shows up. It catches the light from the ROV's lights. And sometimes they reproduce by binary fission. And so they'll still be attached to each other, and they look like those paper cutout dolls where the arms and legs are still attached. Oh, what's this? Just a piece of snow? Maybe. So they look so strange that your eyes are drawn to them. So if they were here, I think we'd notice them. I think they're they radiolarians or theodarians? I think radiolarians. The colonies are quite large, though. We saw quite a few of them in the South Pacific Gyre off the coast of Chile. I don't remember seeing very many here off Australia. Don't know why.
How long's it been since you've been at sea, Dhugal?
February this year, just as COVID was starting to hit Japan. We were the last cruise, I think, that was allowed to go to sea. And then there's been no cruises here at JAMSTEC for anyone for many months. And they're going to sea again now. Everybody gets their COVID tests, PCR tests, and stay isolated for a couple of weeks before we get on the ship, kind of thing. I think you're doing much the same there with the,
yeah, similar story for us too.
Yeah.
Everyone on board the ship has had COVID tests. It's negative. Any new crew that have been coming in to join the ship have been in quarantine for 2 weeks. They come down. So we're in our little bubble here on the ship. We all, well, as sure as we can be. We all know that we're all negative.
So in that case, you don't need to self-isolate while you're on the ship, I would imagine. Like no real need for masks and whatnot, or are you still?
So as long as it's just the...
Tuscarora. Sorry, there's a Tuscarora radiolarian there.
So yeah, if there's been any visitors or the pilot, for example, or anyone from outside coming on the ship, but then yes, it's been masks and precautions and what have you. Once we're underway and we leave the port, then for us on board, it's just our usual hygiene procedures of the hand washing and sanitizing and all that kind of thing. But we're not wearing masks now.
Yeah, because you know everyone's clear. And there's no one suddenly unexpectedly turns up on a ship that far out at sea.
That's right.
No one's flying in from Melbourne. Queensland has very few cases now, right?
Yeah, that's right. Queensland's border has domestic border and international border has been pretty much closed for quite some time. So we've had very few cases all up really, and one or two small clusters that have been well controlled through tracing and lots and lots of testing and that kind of thing. So that's why we've been able to come out on this expedition. All of the scientists that are on board are Brisbane based at the moment. All of us in Brisbane and haven't travelled anywhere. And then later on in the week or into next week, we're making our way up to Cairns, picking up 3 more scientists in Cairns who, again, they live in Queensland and travelled. There's not been the 14-day quarantine needed for any of the scientists.
Right.
Temperatures just dropped below 5. We're at 4.8
great to have
now.
All right, nice. What do we got here? It's another discarded larvation house, I think. Yeah.
Hi Adele on the YouTube, informing us that we just had hail in North Brisbane. I hope my car is undercover.
Wow. Ooh, no, that's just a string, isn't it? I want to get my hopes up. Yeah, this is string. Hail. String of marine snow. Is something? Uh-huh. What is that? Not the squid, no, that, yes.
That looks interesting.
It does, doesn't it? It looks like a siphonophore tail. Which has just been thrown away by the rest of the siphonophore, which is floating up above it. Like the headless chicken with the head still hanging around but not actually attached. I say that is what it is too. Is that the same? Is that the same guy we got a sample of? More shallow, the one that had all the mucus on it.
Yeah, I think maybe it is.
Very similar, doesn't it? Maybe it wasn't still together. Looks to have a lot of palpons.
We've got a question, Dougal. Do squid eat siphonophores?
There's a good question. When you open up the stomach of a squid or a fish, normally the only things you find in there are the very hard to digest hard parts of shrimps or the scales of fish or other things like that that don't digest readily. Jellyfish tend to digest very quickly because they're 95% to 98% water. So when you open up a stomach, you almost never find jellyfish material in there. For people who look really, really closely under a very high-powered microscope, rather than the microscope you'd normally use to look at stomach contents, you can often find stinging cells or nematocysts inside, which point to that the jellyfish are being eaten, but they're just not showing up in most of the stomach content analyses that are being done. And one of the things we're doing here, collecting these jellies, is we're getting DNA barcodes for them. So we can then use those DNA barcodes to investigate the stomach contents of fish and squid to see which species of jellyfish are being eaten by them. Now, right now, I couldn't tell you whether these squid eat siphonophores because there just isn't any information around. But it is known that a lot of different animals, including sharks, eat jellyfish. They're mostly a snack food, like just something, if you haven't eaten for a while and there's a jelly there, you kind of make a grab at it and eat it because it's easy to digest. You know you're not going to have any more yummy food for a little while, so you'll just snack on one until better food comes along. And then there are species that just completely eat jellyfish. There's a species of salmon that lives on almost nothing else. So yeah, I couldn't tell you whether this squid eats a siphonophore or not at present, but I wouldn't be surprised if they snack on them from time to time.
Thanks for that.
There's some really cool stuff done with biologging techniques where they attach cameras, critter cams, to animals, and we find lots of jellyfish being eaten on those. I've been sent images of penguin heads where you see the beak pop out every now and again and eat a jellyfish, or like mola, sunfish, turtles saying, "Can you identify this jellyfish?" And they're normally pretty blurry and not like great cameras, not like the 4K camera you've got here on this ROV. But in a lot of cases, they're eating siphonophores as well as normal jellies, yes. I think it helps that the siphonophores have so many stomachs, so there's a high chance of them having good quality food in the stomachs. So not only do you get to digest the siphonophore material, but you get to eat whatever they've been eating at the same time. Oh, what's this? Looks like a lump of marine snow, but it looks like it has structure as well. Couldn't tell you what it was.
Vicky thinks that looks a
Might
bit
be a
like a
bit.
radiolarian, stuff all over it.
There you go. Does she want a sample of it?
No, thank you.
Oh, yeah, I see. Yeah, it's a tuscarorid. Down there in that massive of snow, you can see a spherical shape there. And the bright white spots there are the single cells. Yeah, I think she might be right. Yeah, cool.
Okay, we'll keep heading down for a little bit longer and then we'll just about be it.
Excelente. Original descriptions of the Tuscarourids, for example, were not of the entire colony that looks somewhat like a soccer ball, but instead they were the patches, like the black patches on the soccer ball, each black patch being one animal, and that's what the species description is, even though when you see them down here in real life, they look completely different to what you see in the books. There are siphonophore species where the original species description is of the tip of a tentacle. That's it.
That's not a lot
so
to
you
go
have
by.
to catch the right part of your siphonophore if you want to be able to identify it. So too bad if you only catch the swimming bells and you try and ID it. You've got to get the part that's been described. So that's where one of these places where these ROV caught specimens, which have all the parts on them, are important, is to redescribe species that were based on damaged material. And it's not that exciting from the point of view of like a taxonomist saying, it's not a new species, we know what it is, this is what it's called, but it's really important to redescribe it so other people can identify it later as we go into the future. This looks like a larvation. We're down around 3.5 degrees, I guess.
4.5 at the moment, Dougal.
4.5. Did that just go back up a little?
No, I think it's been sitting around there.
It's interesting that there's no large scyphozoan jellyfish here, like Coralia or Tiburonia or Deepstaria, Medusa. Not a single one. Just that one Atola we saw with the single trailing tentacle. Otherwise, not even a Paraphila. They're the meaty jellyfish that need a fair bit of energy to survive. So maybe there's a lot of food shallow for the animals that vertically migrate, like squid, but not quite as much food for the animals that don't migrate. I don't know.
Dougal, I'm not sure if you're seeing the comments in YouTube at the moment, but there's a question on there from Anthony, and he says, I heard that octopi are intelligent, but how smart are squid? Any experiments done on those? Do you know anything about that?
Smart as squid. they've got pretty interesting brains. Whether that equates to smartness or not in human terms is a very good question. Like their sensory perception, etc., is amazing on all sorts of different levels. But I don't think one of them would beat you at a game of chess. I think what he said with octopi, you can do things like put a shrimp in a jar, and the octopus will, given time, work out how to unscrew the lid of the jar and eat the shrimp that's inside it. I don't think squid are quite so handy with their arms, pardon the expression. And I don't really know of any experiments where they've shown that squid are smart per se. Like, yeah, again, what is smart? But
smart relative to an octopus, maybe.
Yeah.
So
I think the
not.
animals like octopus or cuttlefish or animals that tend to be able to stay put in one spot and are more territorial. They have to be able to understand their environment and get back to the same hole, or like watch out for danger points and things like that. So they're more likely to be more intelligent in terms of memory than things like squid, which are just short-lived. They only live for like a year or two, or very high metabolism, short-lived, and they range around in open waters a lot of the time. So those kinds of species probably aren't the smartest kids on the block, I would imagine. They just don't need to be. We're almost there.
Yeah, almost
8
there.
meters to go.
0.2 degrees C.
Want that final, wow, look at that jellyfish kind of thing to turn up round about now.
Be right in about 6 meters.
Yeah. It's a lot of big particles. So they are getting food for things that eat snow, that is. The squid haven't turned up for a while.
There was some behind us a couple of minutes ago.
Oh, really? So they're still down the street. Cool.
Yeah, we're a bit over squid now.
Yeah, I'd say squid are smarter than jellyfish.
Yeah, I think that's probably a fair call.
Hello, we got something. Maybe.
Yep, we're just coming in for a look.
So we didn't end up seeing a Pandea rubrum. Where on earth are they? How can they be in both Antarctica and Japan?
Wow, this is so pretty.
Ooh, wow.
Very cute.
4 tentacles, 4 gonads, bright red stomach. What on earth is that? Huh. Wow. Huh. Can you see the radial canals coming out of the stomach? Are there 8 or are there only 4?
Stand by.
Could it be Tetraorchis? That is really cool. Once we
maybe something that's coming down from the bottom, and it looks like there are more than 4.
Okay, sure. This might be a nice finale for that final canister. Stop Google. Ooh, too many tentacles. Ooh, this could be new. Unless there's lots of little tentacles in between those 4 long ones.
Yeah, that's what we think it looks like.
So it's probably Tetrorchis, Tetrorchis, but the gonads look too round to be the species that's known. So it could be new. Please go in, please go in.
I think we got him.
You got him? Awesome.
Oh.
Huh? Maybe.
All right.
That's a yes? Maybe?
Yes, confirmed. We got him.
Awesome. Cool. Alrighty.
We're just going to go a little bit further to get you to 1200, and then we'll call it a day. How does that sound to you?
Sounds awesome. Thank you very much. And I'm very sorry for collecting so many animals when there's so few people on board to do the processing.
There's more people than there were when Jeremy and I first started on the last cruise.
You're a long-suffering battler. Thank you.
It's myself and 2 helpers this afternoon, so we'll be right.
Yeah, and some great piloting too from everybody there.
Yeah,
Very
another
nice.
sterling effort from everybody. Okay, that's 1201.
Okay.
We might just about call it there.
Very
Would you
much.
want to just give us a very, very quick summary of what were the highlights that you saw today?
Okay.
What was
Wow,
there
there's
a new
a-
range extension, or what excited you today?
It seemed there were a lot of heteropods, the mollusk that looks like an elephant kind of thing swimming around in the shallower depths. I hadn't seen that many of that particular kind of animal before in a single dive. It was very interesting. The Colobonema, the rainbow jelly, was at exactly the same depth we find it off Japan, even though we're in a completely different part of the world. Some of the ctenophores, especially that damaged one, looked to be something I'd never seen before. It was really cool. We got some Solmissus specimens and video to try and tease apart the cryptic species question there, which was great. A Siphonophore, which I think might be Maris antarctica, that orange flame-like one with the gooey mucus hanging off it. It would be great to see that and see if it's if it really is that species, and do a redescription of that. that last animal we just caught, Tetrorchus, if that turns out to be new, which I think it might be because of those gonads, that's a possible new species there. It's just been full. Like there's been lots of interesting stuff. Even that fish that we're feeding with would be nice to identify what that is and work that out. The squid, we've got in situ behavior from that species. If we can ID that, that's an interesting story there as well. It's just been all happening, like new stuff all over the place. Great dive.
Yeah,
Thank you very much.
It's a pleasure. It's great having you with us for the dive, Dhugal. Thanks for joining, and thanks everybody on shore for tuning in. We'll have more dives probably later in the week once I figure out what day it is. But not tomorrow. We won't be diving tomorrow. We'll be doing more mapping as we make our way north towards the Great Barrier Reef. So you can all join us again later on in the week.
Excellent. Great. And you'll keep recording this on the way up as you bring the ROV back until...
Yep. We'll keep recording on the way up.
Excellent. I look forward to analysing that as well in the lab later. Really exciting dive. Thanks, everyone.
Thanks, everyone. Thanks, Dhugal.
Yeah, we're still recording. My guess will miss a or two showing up on the way up, so it's also important. You're off.